Alrighty, so I'm assuming you're all able to see my screen.
Yep, we can see that.
Wonderful. So today I'm gonna talk to you about our recent work on using the AI. I'm an applied scientist, so you know, when we talk about AI, there are at least two different ways to work with AI. So you create the models, you use the models. I broadly divide AI associated work into these two extremes: create the model from the scratch, or change the model, update the model, fine tune all kinds of stuff, or use the model, right? So I'm going to talk to you about work that we did where we used the models for our as an assistant for our single cell work. We call this as SCassist. This is a workflow assistant, single cell assistant. So what I actually like to do is, with that introduction, I'd like to actually go in, maybe after a couple of slides, I wanted to jump into the hands-on demonstration and then come back to you for more technical details about the application itself. So we all work with the single cell data. We started with the micro. I remember we used to do a lot of PCRs, qPCRs, and then that few genes, like 10 genes, 20 genes, getting the data out of them became arrays, and we had like 1,000 genes and 30,000 genes. I remember 35,000 genes in a chip. And then it became a sequencing, bulk RNA sequencing. We call bulk RNA today, it's RNA sequencing. And then it became single cell level. So you can assume for every cell, you can assume it as a one experiment. Every cell has about 7,000 to 8,000 genes expressed. So one cell in itself is an experiment, right? So imagine you have on an average, you have somewhere between 500 to 600 cells in a single cell experiment, all the way till like 10,000, 20,000 cells, 15,000 cells. So even if you take about like in the middle somewhere, 5,000 cells, and then they're going to have different combinations of the 7,000 to 8,000 genes expressed. So imagine that the number of data points that you collect from there. It's too much data. You can look at the data in so many different aspects. Every cell is going to be unique. There is not going to be two cells that are identical, right? Because they are technical replicates, or you can even call them as biological replicate. One cell and the other cell is a biological replicate. You're going to have a lot of variations. So you could study those at so many different angles, so many different ways, asking so many different questions, and exploring them in so many different perspectives, using lots of different nice, cool concepts in data analysis, big data analysis. So the data is ballooned that big. It's really the table. If you put 5,000 columns by 30,000 genes, that may be manageable if you look at the face value. But then the questions, like I said, the questions that you can ask, the ways you can explore, that complicates everything, and that becomes pretty quickly very hard to manage with the conventional tools. So let's in a regular standard single cell experiment, this is the normal workflow components. Very pretty standard, right? Very basic. You look at the cells, look at their quality, see how good they are, and understand how the quality is spread across all of the cells for all of the genes, and then you try to remove the bad quality cells, outliers, or cells that express. If there are cells that express 90% of mitochondrial genes, then the cell is obviously dead. If the cell that expresses like 40,000 genes, it is not possible for a single cell to express 40,000 genes. So probably it's because like 3 or 4 or 5 cells, it's a clump, it's a multiplet, right? Doublet or triplet or multiplet. So we wanted to get rid of things like that. So it's possible you might have other, like if you're using the data, if it's coming from PBMCs or blood sources, some other sources, you might have blood cells, other cells that you could you might want it to filter out. You don't want them in your downstream work. So QC, right? It's a very standard technique. You could use the reference-based numbers, like you could have on an average about 8% mitochondria is okay for a normal cell. So anything below that you could remove, or anything above that you could keep anything below that, but above that you may want to get rid of it. So that's one way. That's most that's the most often used approach. Or sometimes folks go review the literature or review the relevant papers, relevant domain of study, and then they identify like 6 or 7 or whatever. The other routine way to figure out the QC is look at the data itself, right? Generate the summary plots, look at the distribution across different samples, and then look at the tail, and you visually look at the tail and figure out where you want to cut off the tail, the outliers. That's another approach. So this is all we've been doing all this time. Similarly, for normalization, clustering based on reference or based on normal reference that's out there, standard reference, or based on your domain-specific reference publications. So 3 starting steps, but there are a lot of steps you go after you do clustering: annotation, differential expression analysis, and then enrichment analysis, and then so that's a standard single cell workflow. And then on top of that, you take these clusters, and then you do really cool stuff, interactions, like how the cells clusters interact, cells interact, how they transition between how the relevance of these clusters in terms of their interactions. Is it possible one is influencing the other cluster, one population is influencing the other population, stuff like that. And then on top of that, you some more cool analysis like a trajectory analysis or the velocity analysis, where you can explore the possibility of the tracing the sequence of a cell from its starting point, or any from a real starting point or a time point, or a hypothetical starting point to all the way particular genes expression to from that point to all other points, all other clusters. Really cool stuff. So, but you also have multi-omics data where you have not only gene expression, but you have the surface specific proteins like cell surface proteins. You can also include the other very routinely done assay in single cell is to include chromatin level information like ATAC. So combining these things along with, now imagine the not only modalities, but also the analysis that you can perform, like the trajectory analysis or the pathway analysis, enrichment analysis. It gets really complicated. It's mind-boggling amount of research space there, that space that you can explore, the data points that are there. So again, the space is, the parameter space is really too much. It's very hard to choose anything objectively. So usually, individuals, based on the bioinformatics expertise, whatever we have seen, somebody has seen throughout their analysis experience, they try to use that, sometimes based on the reference, right? So it's but it's very hard to when it becomes subjective, right? Very hard to reproduce, and there are issues that comes along with that. Also, it takes more than a PhD. It's just with, and I used to say when I did microarray, 2005-6, I used to say, like, it blew everybody's mind. I used to say that if anybody did a microarray, one microarray experiment, like a mutant, I used to work in, I was a molecular microbiologist. I used to work in Staphylococcus. So all I did was growing the Staphylococcus, a ton of them, extracting RNAs and so many different conditions, and then running microarrays and then trying to analyze the data between control and an experiment. I used to say that whenever I used to give a talk during that time, I always said that if somebody did one microarray experiment, there is enough data for you to live your entire research career on that. There is so much data in there that you can explore. So that's what I used to say 2006, 7. But imagine now we are single cell space and the tools and the opportunities that we have computing new concepts. It's really going to take more than a PhD, and it's also when I say more than a PhD, now I could say either an individual human, a person, or a PhD level AI assistant, right? So you could have an assistant, but you need that much knowledge and depth into your field for you to explore any of these things. We are only scratching. Even it's even harder to scratch even the surface. You see, so much the parameter space is so much there. And absolutely, you'll need a lot of coffee to work on this. So I used to say, again, microarray, I used to say it's a data ocean. Either you get drowned or you figure out how you want to navigate it. So again, it's the same thing. We are still the same data ocean. If if I saw this as data ocean microarray era, like 2006, 7, imagine what we are into now. Yeah, you definitely need a lot of coffee so you don't get lost in the ocean. You don't lost in the forest. Yeah, all right, and keep yourself enthusiastic about what you're seeing. All right, so that is the big data single cell situation we are now, and that's what, like 2 years ago, this is all I was doing. I was doing single cell, a ton of single cell data analysis for my colleagues. Now I'm a computational biologist. I work closely with lab folks, and especially I work on at NEI. I work on a bunch of autoimmune disorders from the coming from the laboratory of immunology, mostly autoimmune disorders. We do a ton of single cell experiments. And my main job was to help the folks starting from designing the experiment all the way till publication, preparing the data and submitting it, the fair concepts and NIH data management policy stuff, analyzing the data and stuff like that, right? So 2 years ago, right? I started like 4 or 5 years ago, but about 2 years ago is when LLMs come into the play, the large language models, or the so-called AI. And so, you know, I tried using the models like everybody, and then it quickly became, working with the chat window is not possible for so many reasons, because the amount of work that we do, the amount of questions that you wanted to ask, it becomes really, and to be able to navigate and manage that chat becomes very quickly, it becomes like the worst way of handling your situation. So I had to explore the option of accessing the models and the power for analyzing single cell data using programmatic approaches, right? So most of us end up usually in that way. So I started using the R as my picked R as my the main platform. Python is popular and famous. R is also is next in the line to Python, even in the data science, that. So I chose R because at that point, like 3-4 years, Surath is all we were using, Surath and ScanPy. But Surath mostly, so the main Monocle, Surath, so everything I had to stick to the R platform. So I chose R, and I worked on this integration and using programmatically or automating this process, whatever I was trying to do, my single cell data analysis, right, using these LLMs. And then it came to a point where I wanted to, I thought this will be a good, from input from my colleagues, we thought this would be a good tool for others as well. So we put together this nice tool. And then we have to go through all this routine LLM-based research approaches, like evaluating them and testing them from all perspectives, right? So that's the bigger picture. And I like to show you the tool now very briefly. I wanted to demonstrate it and then go into the more details about the tools, the back end of the tool, the results that we got and stuff like that, evaluations. Are there any questions from the bigger perspective point? All right, there are no questions. So let me also go through this very quickly. LLMs, large language models, AI, aka ChatGPT. Its counterpart from Google is Gemini line of models. Meta has Llama models. X has Grok, and you have Claude. You have ChatGPT is from OpenAI. So you by this time that they literally can do almost everything. I don't know if there's anything that they cannot do. There are some things that for now, they're not able to do. Maybe for a foreseeable time, they may not be able to do certain things, but they literally do almost every mundane, routine, regular, day-to-day things that we do. They're able to do it pretty nicely, pretty sophisticated way, pretty easily, pretty beautifully, mind-boggling way that normal human beings may not be able to do it. In the biomedical applications, they can not only we've been analyzing the data, but now LLMs are able to infer the data. They can actually, I don't know if I can say, if I'm allowed to say the word think, but kind of like that. They're able to connect the dots. I can put it that way. If thinking is not is too much, but they can connect the dots pretty beautifully. Dots can be connected the way that you and I cannot, or at least I cannot connect the dots, see what's in the data and connect the dots. Well, I don't know if we still need the PhDs anymore, but I'm just assuming that there was an article. I don't know if you're on LinkedIn. I had a post like 3 months ago, 4 months ago. There was a study about how students' brain activity are between two groups. One that writes essays based on using GPTs, and one that does not after using GPT, right? So it's you can see two ways. It's scary that your brain tends to not fire up much as using ChatGPT. When you don't use ChatGPT, you could go to that side of conversation like it's scary, or you could go the other way where you could consider that as an opportunity. Now that your brain is not firing, but so that space is now going to be used for something else. So what is that something else might be? That's a evolving learning modality. So what that could be, what is that going to lead us to? So that's a positive, and we don't have a choice. So two different. So if you're on LinkedIn, check out the article that I posted. It's not an article, but it's asking questions on another article that talked about the AI influence on the actual neuron firings data. So do we need a PhD anymore? We might need PhDs. Yeah. The other stuff with AI that we often talk about is hallucinations, meaning they make up stuff. They make up stuff. There's argument that it's a bug, or it could be fixed, or it's a feature that's always going to be there. We just need to learn how to handle that. It's impossible not to, like we all make stuff up. So, it's AIs make stuff up. But there are approaches that people have put forward. RAG used to be one of the popular ways to, it's a separate a component in a infrastructure, in a framework that can ground the AI by providing relevant data, not letting it loose to go all over the place. So SCassist, in our application, we didn't actually use a RAG itself, the textbook definition of a RAG, but something similar to that. We call that as an augmentation, prompt augmentation, something like RAG, but not identical. So this is about the LLMs. Let me go. So having understood the LLMs, and then our existing problem of the single cell, my trying the chatting windows, and very pretty quickly coming to a wall, hitting the wall that this is not the way that I want it to work. Very inefficient. Then hooking up the Gemini to the R, and then trying to integrate it, and then building that into an application standalone that everybody could use. So, right, that's a bigger picture. I'd like to now demonstrate SCassist before we dig deeper. And I'm going to fire up my RStudio. Please feel free to interrupt me if you have any question, if you wanted to comment anything that along the lines that I'm talking about, or anything that you disagree, please feel free to chime in. All right, so here is our
Yeah,
RStudio.
so you can either say put your question in the Q&A session, or you can raise your hand, so we will unmute you if you have question. Thank you.
Absolutely. All right, so here's our R. So I'm going to go to, I'm going to search for SCassist, and I'm going to go to the GitHub page. So that's our repository. And what I'm going to do is I'm going to go down all the way to the installation. So I have the requirements already installed in even the application itself, but I'm going to, for demonstration, I'm going to show this. So I'm going to copy that, install, and it's going to complain. It's going to say that I've already installed it, so I can force it. So as you can see, I've already installed it, but I could still force it to reinstall it. The install should be pretty quick. Asking for my updates. I'm just going to skip. I don't want any updates. It's almost there. So right after its installation, what I'm going to do is I'm going to run this example usage. I'm going to load the libraries. So SCassist Surat is our library for single cell RNA-seq analysis. I'm going to download. Here is an example data. It's a H5 format data matrix. It's a the feature barcode data matrix. It's just one sample, just for demonstration purposes. And we have used this data set in our publication for evaluation. So if you're interested in exploring that more, but I will download this H5, one single cell RNA-seq data. I will load these two libraries. And then, so this is the standard, this is our input data, and this is the standard Surat way of reading the data. So I read the data. It's a 10x genomics-based data into an object called KO knockout. And then I use that knockout, and it's got multiple modalities. I think we have ATAC as well. So I'm going to extract only, create a separate assay for gene expression alone. I'm using the Surat create Surat objects function, and I'm going to store it in the KO object. So these two are standard Surat commands and Surat workflow components. So you can see SCassist, it says done. Our installation went through fine. And I'm going to load these two libraries. So SCassist is loading now. It loaded, and then I'm going to load Surat. That's also loaded. Move this up a little bit so you can see. All right. So I have already downloaded this. Just by clicking on this link, I should download that automatically. It's just direct link to the file itself. So I'm going to read that file now. And it is in my downloads folder. So I will fix that. Downloads, and then so that's my file that I just downloaded. So I'm going to read that file. And while that's reading, you can see it's reading the gene expression alone from the multiple modalities file. And then I'm going to create the Surath object. So that's it's done already. And you can take a look at the Surath object. You can see it's a Surath class object. It's got about 32,000 genes in total and about 4,800 cells in it. And we're only working with one assay, which is RNA, the RNA expression data. And we have not done any analysis on that. So that's why you see no variable features, no PCA, no nothing, right? It's the raw data. All right, the matrix, count matrix data. All right, so let's go ahead and the next one is to, I need to set up the API keys. And I have links here for you to follow Google API. If you want to use Google or ChatGPT, these links will walk you through step by step on obtaining the API keys. So assuming that you have your API keys, I have to specify the location of the API key. So I'm going to specify the location, just the location of the key file name. I have it in my downloads. So I go like that, and then API keys. That's my Google API key. So I run that. So I have set up the API key file. And then here is my first command. I'm going to run the QC. So what we are doing here is we're calling the function. It's a SCassist analyze quality. And I'm going to use the Surath object .ko. And I'm going to specify the server as the LLM server or the LLM service provider as Google. And I have to specify the API key file, which is what it is here. And you know what we have in the KO. And the only other thing I have to do is the LLM server. There are a lot of other parameters that you can specify here. There is a whole bunch of parameters you can specify. For example, you will see now when I run Google, SCassist default when it was when we developed it was Gemini 1.5. Gemini 1.5 doesn't exist anymore. So if you run it, the default, if you run it now, there's not going to be anything there. So the output is here. If you run this output, you will see it's null. There's nothing there because it's running the default model, which is 1.5. You could actually, I think we might have it in the documentation. So how do we specify the model, right? So if that's the default 1.5 it's deprecated, how do you specify the latest model or choose whatever the model that you want? So one way you can do that is first look up how to, you could actually start typing here model, and you will see the SCassist as helping you with the parameter model for if you're using Google as the server, then model G is a parameter. If you're using the local version, model O. If you're using ChatGPT, model C. You have to, that's the parameter you specify. You can also look at the actual documentation. SCassist analyze quality. And that should pull up the documentation here. You can see this is the default model. It is 1.5 flash, but you can change the model using that parameter. And there are other parameters that you can specify here, including the model parameters. You know, if you want to change any temperature or any other, whether you want to have a thinking or non-thinking or a specific, the thinking, how much, what level of thinking you want, that kind of thing. All model parameters could be specified here, right? Okay, so now that we know how to specify the model and 1.5 has been deprecated, so let's go ahead and find out what model we want to use. So one way you could find that is search Google models, Gemini models, or Google models. Either one should work. So go here, the first one. So here you can choose 2.5. Pro is a lot of money. Then there is another place where you can check the prices, compare the models. But Pro is it's a little bit costly, and even the Flash, the cheapest version you can get is from the light version. It's faster, It's a small model, as well as it's the cheapest one. It's a cost efficiency, as you can see. We have shown in our publication that the models themselves different models do not have any statistical difference. That's simply because the way that we are grounding the LLMs, right? So if you expand this, you will see the exact endpoint that you want to use. So you could just copy that, and you could specify model. It's Google, so model G equals, and I put in the model name. Now, if I run it, it might take a few seconds. You will see the results already here. So I don't want to read this, but just to highlight, first thing it's going to give you the reasoning, right? So there are only two metadata columns that we have. So hold on. So by the way, let me show these metadata columns. So if you look at the metadata columns here, we only have two metadata columns: number of RNA counts and the number of feature counts. So these are the number of genes. These are the number of actual transcript counts. So it's going to take whatever is available. If you have other metadata columns, for example, if you have hemoglobin percentage, if you have mitochondrial percentage, if you have ribosome percentage, any gene-specific percentages as metadata, it can use that. And you have option to specify those additional columns as well. All right. So now that because
one question, Vijay.
Yeah.
Sorry to interrupt here. So the model you are using right now, is that a free one, or we have to pay for that?
That's a great question. No, it's not a free one. So all the models, the only model line of model from Google that's free is Gemma.
Mm-hmm.
Gemma is the only free open source model. All other models are if we go Google Chat, the public chat, you have some free things that you can do, right? But otherwise you pay for monthly. But this is we are going through the API calls from the backend, so they are dirt cheap. So you have free quota, right? The model itself is not free, but you have free quota. But most of the time, the analysis that we do in the paper, that we show that for 2 months of work using Gemini API calls, all I ended up was $2.
Mm-hmm.
So I probably about 1,000 or 2,000 calls. I ended up only $2.
Great.
Did I answer the question?
Yeah, thank you.
Sure. I will quickly show you the Llama version, local version. It's completely free. All right. So you can see I showed the two columns that we had. Now you can see what it does. It's basically taking that. It's SCassist LLM, right? There are two components, right? Surat, SCassist, and LLM. So Surat has these numbers already, and what SCassist does is it calculates the summary data for each of this metadata column, and then it feeds that summary data into the LLM, right? So that's where the augmentation comes in. That's where the grounding comes in. That's where the RAG idea comes in. So now the LLM is supposed to answer or infer the quality of the data and provide suggestions based on the summary data that we provided, right? So it's already grounded in the summary data. So that's where SCassist comes in. There are additional components. It's not just sending your data directly to LLM, but the prompt is augmented with data that are computed by the other components of SCassist. Now you can see it's giving the reasoning why, what is the cutoff point that you might want to use. The lower cutoff and upper cutoff, and it gives you a recommendation, right, for both the features and counts, and then it gives you a final summary. This is what you want to use for the lower and upper cutoff. All right, so this is you can, if you remember, if you are able to remember, you can keep this in your mind. This is 1,200, 2,800, 700, and 5,000. Let's run the same analysis using ChatGPT, right? So now I don't need this. Even if I have that, it's not going to change anything. So all I have to do is change this Google server into it. I don't know the actual parameters, so let me take a look. So the model, Google or OpenAI. So my values can only be Google, OpenAI, or Ollama, right? So I'm going to ChatGPT is OpenAI, so I put that as my server. and I need to change my API keys, the key that is specific for OpenAI. So I have another key that's specific for OpenAI. So set that up, and then again, the results are displayed interactively to the user. It's also stored in the QC recommendation, so you can take a look at that as well. So this is the you could neatly, if you wanted to take a look at that. So it's also stored in a variable that you could use later. All right. So I could save this as QC Gemini. And then we can run this now again using OpenAI. So let's see how that looks like. All right, so there's your numbers and similar ideas, similar concepts, LLMs. So it gives you the reasoning, its analysis perspective, and then it gives you the recommendation. You can see the summary: 1500, 500, 1200, 4500. Not that dramatic, but similar, very similar. 1500, 500. You can see 1200, 2800, right? So you are going to see some differences because they're LLMs, individuals, right? You consider them as like individuals, but pretty close to each other. Okay, so let's now run using Llama. So I'm going to change the server to Llama or Llama. Again, I'm copying this from getting this from the documentation here. I don't need Llama is a local server. I don't know if you can see Llama is running here for me. I'm already running Llama locally. Llama does not need internet. It is not connected to any Ollama. Ollama is an application that runs open source models. It can run open source Google model, which is Gemma. It can run open source DeepSeek model. It can run open source Quen. It can run open source Llama. Right? It can run any open source model. It can run your own model as well. So I'm running Llama here already. And I already have set up, downloaded. You have to download the model, whatever the model you want. And there are hundreds of models that are available. So if you are interested in any of the models or testing it, so if you go to Llama models, you can see all the GPTs, Gemma, Quen, Llama, Nomic, everything is here. So whatever you want, you could download and use that model. All right, so I have already downloaded Llama 3, which is the default for Ollama. So you can see if I specify the LLM server as Ollama, it's going to use model O as Llama 3. But you can always specify, change it. Llama 4 is powerful. That's the latest. But Llama 4 takes a while. You might need, because this doesn't go to server, you can work if you're worried about security, if you're worried about confidentiality, if you're worried about not wanting to share this data with anybody else, you can cut off the internet and run Llama locally. It's a local server. It is run locally in your computer. The Llama 3 is faster, super fast, and Llama 4 is a little bit slow. I'm saying faster and slow for a out of the box commercial or a personal laptop, right? But if you have a bigger machine, Llama 4 might be faster as well. But I'm talking about regular, like a 4 gigabytes or 16 gigabytes RAM machine. But you're not going to see any humongous difference, statistically significant difference between Llama 3 and Llama 4, simply because, like I said, they have the basic knowledge, 1 or 2 years difference in their knowledge cutoff, but they are grounded the same way with our data. So because of our grounding, and majority of the data knowledge is already there, captured in Llama 3 and Llama 4, you're not going to see any statistically significant result differences. So I would encourage Llama 3 first, and then you go from there. So if you run Llama 4, you might get disappointed because of the time that it takes. All right, let's run Llama and see how that looks like. So before that, I want to save the other data. So this is, so that this will be ChatGPT. So I'll save that, the current QC recommendation as GPT. All right, now let's run Llama. It might take a few seconds more than your Google and ChatGPT. Google is the fastest that I've seen, but not much difference between Google and ChatGPT. Llama might take a few seconds, but Ollama might take a few seconds. And also while you run Ollama, you will see your computer making the sound, the fan starting to spin faster and the louder noise. Might take a few seconds, that's okay. But so when you call Llama for the first time, it's going to take a minute probably, but then the subsequent calls are going to be faster because it loads everything into the memory. I can hear my computer fan speed picking up.
Hey Vijay, we have two questions in the Q&A session. I'm not sure you can see that or not. Would you want me to read that for you?
Yes, no, I'm seeing it. Thank you so much. So the first question, Lauren, a great question is Llama 3 free? 100%. Yes, all the Llama models are free, open source models. That's a big difference between Meta, Google, and ChatGPT. I'm not sure, top of my head, if OpenAI has any completely open source version of GPTs, but Google does have Gemini, which is completely open source, meaning completely free. Free as in free beer. No, you can do whatever you want with that. Likewise, Llama 3 is free, Llama 4 is free. All the Llama models are free, open source. Absolutely. Okay, that's the first question. I hope that I answered your question, Lauren. The other question is from Maya. Can you use the free version of ChatGPT? I think you're talking about the ChatGPT Win, the software. Online, we go and put in that thing, right? So the free version, like I said, the online chat, the free version we are talking about, it's like a free quota. You can only do so much in there, right? So, but we are using the chat GPT not from the GPT window, the chat window, but from the API, from the backend. So using the backend, you do get that free quota. So it's not a free version, but there is a free quota. I think the version that we do is also it's a what do you call the low end version of the actual GPT. I don't know if I answered the question. We are using APIs, not the ChatGPT window, but if you use run ChatGPT for SCassist, I'm going to assume that if you're not a 24/7 bioinformatics person, if you're running this analysis for your research for one experiment or two experiments or even for 10 experiments, I think your calls, API calls, will fall under the free quota. And even if you get charged, it's like I said, 2 months of our research for this SCassist cost us only like $2. The API backend is, even though the front end, the GPT, the chatting window, it's costly, like $20, $30 per month. But APIs are pay as you go. So there is no upfront fee. Only if you call one call, probably 10 cents or 2 cents for every call. So it's pay as you go. It's Maya, I hope I answered your question. If not, please let me know. All right. So our Llama results are here, as you can see. This is all completely free. Llama 3 gives us that it's going to the safer side, 500 compared to 1,500 for the others. But the higher end is 20,000, where we saw 22. And the other for the feature counts 500 and 4,500. So pretty close, precisely like ChatGPT. And it's giving us a little relaxed threshold. Rather than 1,200, it's asking us to use 500. So you see the difference there, right? All right. So that's all I wanted to demonstrate. There is so much we could do here. I wanted to quickly go back and show you a couple of other things you could do, cool things.
Sorry, we have one more question. Sorry, Vijay, we have one more question from the audience.
Let me go to
Ziyang, let me just unmute you. Okay, I think now you can talk.
Hello, can you hear me?
I can hear you, yes.
Yeah, hi Vijay. Thank you so much for this good presentation. I have a question,
Yes.
and I want to know whether does SC assistant finish the post train after the maybe any large language model, the post train.
Not sure I understand the question. Could you repeat it again, please?
Yeah, we know the language model can answer many questions. I think most of them are general, maybe not specific for biology problems. So my question is whether the tool of SC assistant do the post-train after the language model, such as LLama 2, LLama 3, and so on. Yeah.
So are you asking if we can train the model Llama 3 with the single cell data, or?
Yeah, because the SC assistant tool can give the correct or good answers about the biology problems. I think the post-train is important for the tool. So I want to know whether you have done the post-train. Yeah.
So you mean post-train is what you mean? Are you talking about fine-tuning?
No, post-train is between pre-train and fine-tuning. I think the whole phases is pre-train, post-train, and fine-tuning. There are totally three phases, three stages. Yeah.
So there is zero training in our tool, right? SCassist, like I told you in the very beginning, there are two classes of groups of folks that you can approach LLM use, right? So what SCassist we have done is as a user, you applied purposes, right, for the model itself. But I would agree with you that post-training, even pre-training or fine-training, any different trainings, we could study, research, and do some trainings. And there are fine-tuned, not fine-tuned, there are even pre-trained models, foundational models, SC, I think it's SC GPT. There are a bunch of other foundation models, single-cell models. I'm pretty sure there are other fine-tuned and post-trained models too. But I'm pretty sure it will be of help here. It might increase the way the better answers. It might help us get better answers. But given the way we are grounding the LLMs in our data, right? And there might be some improvement, but I would doubt any statistically significant changes in its response.
Okay, I see. So the SC assistant is using the LLama 2 or LLama 3 directly, right?
Say that again, LLM.
So it just use it is just the direct use of language model, right?
Exactly. Exactly.
Okay,
So,
I see. Okay, yeah.
Absolutely. Yep.
Okay, thank you.
Sure, you're very welcome. So there is not even, I mentioned RAG. So we're not even using RAG. What we're doing is the main idea is augmenting the prompts with metadata that we compute, right? And that's the only trick here. Relevant metadata, hooking it up to a single cell Surath object, generate metadata, augment the prompt, use that augmented prompt to query the LLM. So that is the main brain. All right, wonderful question.
One more,
all right, let's see.
One more question in the Q&A session. Lauren.
I think I answered Lauren's question.
There's another one.
Yeah.
There's another one. Does this work for very large data sets, like for example, 300,000 cells?
100% yes, because so what the things we are doing, right? The QC is based on the QC is simply based on the summary statistic, right? So it doesn't matter how many cells we have, right? It's out of the way. And for example, normalization might take few seconds, few additional, because for it to recommend the normalization, it has to, the metadata that's calculated is based on all of the data. So it might take a few extra minutes, but I do not see any bottleneck in terms of the number of cells that SCassist would be affected by. And the other, for example, the variable features, it's simply based on the number of features that are in the variable feature slots. It's not based on the number of cells. So this will not be affected. PCA, again, it's not running the PCA, right? The PCA is run by the Surath itself, and it's using the PCA slots. So because it's using the PCA slot, it's not affected by the number of cells. It's only affected by it's going to be the same number of PCs or the same number of genes on an average in any number of cells. That's not going to be affected. So finding neighbors, again, after you find the clustering, the clustering might take few minutes, because for it to recommend the K number, as well as to recommend the number of clusters, some of the standard deviations and stuff that you calculate, mean expression stuff like that, it might take a few extra minutes, but I do not see that as a bottleneck. Again, the last one is the annotation. Annotation is fully based on the top genes in your find all markers results. So it's not based on the individual cells, but the metadata column itself. So I don't see any of the functions, the number of cells being a bottleneck for any of these SCassist functions. I hope I answered that question.
I just kind of have a gentle reminder. We have 5 more minutes, Vijay.
Absolutely. So what I'll do in the last 5 minutes is to show you some of the things it can do. Annotations with SCassist. You could do annotations, like for example, could do nice pathway enrichment analysis. If you have within R, could put together all the enriched pathways into a nice networks to give you higher level. Look, it could do KEGG analysis. It could do ontology on a geo, and it can put together. It can integrate both of them and can give you a nice summary, key genes, potential targets, and stuff like that. And I wanted to show you the annotation part. It's something I've not seen any other annotations tool do, which is, let's see here. It'll give you reasoning for why it's here. So if it calls the cluster 0 as neutrophils, it'll tell you why it called cluster 0 as neutrophil. It'll give you all the main markers that are in that cluster, and that's the reasoning why it's calling. So I'm going to stop here. Maybe the 3 more minutes, I'll let, if anybody has any questions, I'm happy to answer.
Hey, Vijay, one question here, since you talk about the cell type annotation, use the SCassist, right? So what about the accuracy? Did you test the accuracy for, to use the SCassist for cell type annotation? I mean, for ChatGPT, right? Usually, if we just put in, like, for example, the different expressed genes for each cluster in ChatGPT, it will give you some annotation. But what about the accuracy? Did you have a chance to do some study?
That's a great point. We did not do any benchmark on the accuracy of the annotations, but there was a reviewer that asked a question about, can you at least compare with another tool? So here we show in the supplementary GPT cell type. It is similar to R-based. So it can run, it will take your markers all, and it runs GPT. It will take ChatGPT like SCassist, but this is the output that you get, right? For the same data set, you could see SCassist giving a very similar cell types, but the only difference is it'll give you the reasoning behind why it called these cell types. So we compared these annotations from here with this GPT cell type with the SCassist. So, and we show that it's pretty much very similar. We didn't quantify it, and we have not done any benchmark.
Okay, I think Yeah, we have one more question from Lauren. Can you define what cell types you like? I don't know. You would like annotated?
Absolutely. So there is a beautiful, that's a great question, Lauren. So there is a nice, very nice option here. So when you run SCassist, right? So there is an option that if there is a parameter, I'm searching for this experimental, searching for it here. There is a beautiful parameter you can set. It's called experimental design. So you can specify not only the cell types that you're interested in, but also, I'm showing here it's a single line PBMCs of one healthy control versus one uveitis patient, right? So you could put in other things, questions that you wanted to ask. You can the other things that you want the LLM to focus on. So that will get appended to the augmented prompt, and that will go along to the LLM. So your inferences will be based on the experimental design. Absolutely.
Okay, we have one more question from Monica. Let me just unmute you. Okay, I think now you can talk.
Hi, thank you, Liang. Hi, Vijay. So my question is, sometimes Sarat's integration and things may not be ideal. So do you think I can work with Harmony and other packages also, or is it just restricted to Sarat?
So it depends on what function we are talking about, right? So if you integrate using Harmony.
Yes, integration.
So if you integrate with Harmony, then what function are you thinking about calling in SCassist? What is that because SCassist has about like 9 functions, right? You can do 9 different things. So
I see.
yeah, so after you do integration, I'm going to assume that if the integrated data set is in the Surat object again, right?
Yeah, I guess we could convert it back to a Sarat object. Yes.
But for now, if you wanted to use all of them, then that's the only way.
I see. Okay.
Again, depends on what function you wanted to use.
So yeah, I think primarily I was thinking sometimes I just don't use Sarat, like I
understand.
So that was my primary question, but I think I understand that if I just convert it back to Sarat, I can just use this. So yeah, thank you.
That's the only option for now, but that's a great question because you asked it. I did not meant to say this, but I wanted to say that we have the version 2 that it can SCassist can work with cell chat objects, and it can work with monocle objects, and so we potentially may be able to integrate it with other objects, including Harmony.
Sounds great. Thank you.
Absolutely. Any other question?
All right.
Yeah, there are no questions. It's wonderful meeting you all. It's a pleasure talking to you, Liang, and I think thank you so much for the opportunity.